Hello guys, very welcome back. In this video we'll be learning about cloning, artificial fertility technique, okay, which is done in female and which is done out of female, okay, that is in womb and out of womb. And finally we'll be learning about what is stem cell technology, what are these cells, and what is the stem cell therapy and the type of stem cells. Okay, so let's start with our first topic, cloning. Now, clone means copy. Clone can be defined as a child of single genetic parent. Okay, single, that is either mother or the father, right? Now, clone can be made by a procedure, very important procedure, and this is the single most important term in cloning, that is somatic cell nuclear transfer. Let's understand this word, somatic. That is, in cloning you only need somatic cells. What are somatic cells? The bodily cells, for example the skin. Nuclear, see, we need all the 46 chromosomes of the skin cell, because we are cloning the person. We are not here to get half of chromosomes from father and half from the mother. Since we need all the chromosomes from the somatic cell, hence nucleus of the somatic cell is taken out and the nucleus of egg is removed and the nucleus of the somatic cell is put into the egg's nucleus. Okay, what is left is the cytoplasm of egg surrounding the somatic cell nucleus, and this is known as the cloned zygote. Now once this zygote is formed, it is transferred into the womb of woman, okay, and hence after 9 months we get a new cloned body, right? The next is artificial fertility technique. Now this technique can be used both in the womb of a female or outside the womb of female. Okay, so let's first understand what are some of the reasons why we are going for artificial fertility, right? Okay, see, man or the female or the woman can suffer from many fertility issues. For example, there could be azoospermia, where there is absence of sperms in man. There can be oligospermia, where there is few or less sperm. Okay, so we make cell banks. Okay, for example, the sperm banks or the egg banks, right? And these sperms and eggs are kept in and preserved in liquid nitrogen. So cells are preserved up to 100 years. So these were some of the reasons. We'll discuss some more reasons later. So let's start with the fertility technique. The first technique is in vitro fertilization. Vitro means lab. So in vitro means in a lab fertilization. That is, fertilization is done in lab. Let's see closely. So egg cell is taken, or sperm cell is taken, or both are taken. They are fertilized in a test tube. The resulting zygote is transferred into womb. Next, ETT, embryo transfer technique. Same technique, IVF, but only the difference is zygote is not transferred, rather it's the embryo, which is after multiplied is transferred into womb, so it has better chances of success. Next is IUI, that is Intrauterine Insemination. Okay, now in case of oligospermia, where the sperm cell is unable to travel and reach the egg cell, the sperm can be collected artificially here, and these are injected at the site of the cell. Okay, now this drastically increases the chance of fertilization. Now this is used in for artificially endangered species. Okay, these all techniques can be used to increase the birth rate and avoid them from extinction. Okay. Now, one beautiful example of this is that now when the north white rhinos were about to get extinct, the southwest rhino was used as a surrogate. Okay, and the north white rhino was prevented from extinction. Okay. Now this all this technique in case of human requires the transfer of this thing. At least first two requires the transfer of the either zygote or the embryo into a womb, right? So this is known as surrogacy. That is, the process of lending uterus is known as surrogacy, and the womb which is used is the lady is known as surrogate mother. Okay, so we have still surrogacy regulation bill pending to become an act. So surrogacy regulation bill 2016 says that the couple out to be an Indian, they must be married at least for 5 years, and at least one must be infertile. Okay, it should not be like both of them are fertile but both of them are not willing to undergo the natural process. The surrogate mother should be close relative. It should not be a stranger. There should not be any commercial transaction except the medical expenses covered. And the surrogate mother must itself be married and must have her own child. And the surrogate mother can't claim the baby. Now, in case of termination, the consent of surrogate mother has to be taken. All right, that's all about the in womb transferring. Now, out of female. Okay, now we have the concept of biobag. Now, in biobag, the natural womb condition is simulated. For example, the electrolyte solution against the amniotic solution of womb. Oxygenators are used for the constant oxygen supply. Nutrient supply is maintained. The constant fluid exchanges so as to remove the waste. Okay. Now, in humans, this decreases the neonatal mortality. All right, that is death of child immediately after the birth, and extinct or critically endangered animals can be saved. In fact, talks are going in even for dinosaurs to resurrect them. Okay. Now we discussed cloning, artificial fertility technique. Next we move to stem cell technology. Okay. Now, what are first of all stem cells? As the name suggests, these are the cells which can be converted to any other cell type. Now, after all, why we are going for this technology? Because organ donation has lots of problem. It's very difficult to get a suitable donor. Even if you get your siblings to donate their organ, the average matching of chromosomes is only 75%. Because and that too, if you have more than same parents, then the chromosomes match more than 50%. That's for sure, and less than 100% because of the recombination differences. Now let's study the stem cell therapy, right? But before stem cell therapy, we need to understand what are the types of stem cells. Okay, so the first is totipotent precursor stem cells. These are the cells that can be converted into all other cell types, all the body cells, all the somatic body cells, umbilical cord and placenta. Zygote. Pluripotent precursor stem cells that can be converted into almost all other cell types. Okay, but not placenta and umbilical cord. Embryonic cells. Multipotent precursor stem cells that can be converted into many other cell types. Okay, umbilical cord. Oligopotent precursor stem cells that can be converted into only few other cell types. Example, the bone marrow cells. Okay, that's why they are used in different blood cells. For example, in blood cancers, this bone marrow cells is used. Okay, even in leukemia and other blood diseases they are used. And finally, we have induced pluripotent stem cells. Now, if you see the somatic cells, its ability to convert into organs, it is silent. Okay, but when the somatic cell is taken and the embryonic genes, it's activated by the embryonic genes. So, for example, the SOX, OCT4, Lin28, and Nano-G, right? So it also it becomes induced pluripotent. Now it is able to produce all the body cells, and for this, Shinya Yamanaka got the Nobel Prize. So that's all. We discussed about cloning, we discussed about artificial fertility technique, and finally we discussed about stem cell technology. Thank you.